these videos into YouTube channels and putting it on a YouTube channel. So if you miss something or you know somebody who has, there'll be a YouTube playlist for this class, so you can go back and watch what we did in class in case you need to see it again and kind of watch that over again if you're a visual learner like I am. All right, so today we are going to be starting with DNA structure, function, and replication. So last week we talked about the cell structures and functions and how passive transport and active transport work. Today we're going to start talking about DNA. I know we're skipping around a bunch because I'm just trying to get a condensed summer school version into this class. So first up, facts about DNA. It stands for deoxyribose nucleic acid. Okay, deoxyribose nucleic acid. And those things should pop up in color. So that goes deoxyribonucleic nucleic acid. It is too small to see with the naked eye, but under microscopes it looks like a twisted up ladder. They call this the double helix. DNA is a code or recipe for forming all the proteins in your body. So proteins is not just the stuff you drink when you're trying to gain muscle. It's actually codes for structures that tell your body and the things inside your cells to do certain functions, whether it's kill a certain cell, or it's to duplicate, or to split in half, or to attack a certain cell. These proteins are kind of like messages that go back and forth. So DNA is a recipe for that. It tells, so the DNA inside your skin is a recipe for how to make a skin cell. So when you get a cut, your body closes the cut, you're forming new skin cells over top of it, and it's replicating this in stuff. So instead of when you get a cut, you're not forming an ear on top of where your elbow should be, or something like silly like that. So those are codes or recipes for forming proteins in your body. So I'm filling in the notes along with you guys. So there's some pictures up in the top corner there. It kind of shows you a simplified version of what this looks like. We're going to be diving into why they look like that and what those purple, green, yellow, and red things are too. All right. Proteins are macromolecules. If you break down the word macro, macro is like large, or talking about the whole picture of something. A molecule is anything larger than an atom. So it's combinations of atoms that can make up molecules. So these are large molecules. Basically what that means. All right, so if you're following along, you should have had the first two parts of the notes complete. All right, so quick facts too. These aren't on the notes, but I just want to let you know. Every living thing has DNA. That means you have something in common with a zebra, a tree, a mushroom, a beetle, an ant. Anything that is living has cells and that has DNA. We are related to in some way, shape, or form. That's a long topic, going back to the first ancestor that crawled out of the ocean a long time ago. So that kind of stuff, we actually do have something in common with all these animals, plants, and trees. All right, so genetic material, DNA, must, so in order to be determined to be DNA, it must store information that can be passed from one generation to the next. So you must be able to pass on instructions for how to make yourself. That's like key to being considered DNA. All right? You must be able to copy yourself in order to pass on the information. And then you must be able to be strong enough to sustain mutations. So mutations are not always a bad thing. Mutations can be good things. Your DNA mutates when it copies all the time, but there are things in there that stop the mutation from becoming bad. If it is bad, sometimes mutations are good, or they can be harmless. So not all mutations are bad things. You might have heard of, oh, the DNA mutated, or it might be looking bad, or mutations in strands. Sounds harmful, but it's not always the case. Kind of with what's going on with COVID right now, the COVID-19 virus is not mutating, which is good for us. The more it mutates, the harder it is to nail down a vaccine for it. So the more mutations of a strand, that's why they say the flu shot doesn't work. The flu shot is still a good thing. It covers you against flu in a lot of mutations, but there's so many people have it and it mutates so much that it's hard to nail down one specific thing. Okay, so you must be able to have these minor changes, which produce variations in the population, which are why you don't look like me and you don't look like, you know, other people in this classroom. And that's what the variation is. It's how we're all different. Okay, so I'll give you a second to type that up before I move on. Some mutations can cause the cell to be unstable and then it'll die off, which is bad. So that's a lot of how a lot of diseases happen too. Is mutations that cause harmful things to occur. That's what cancer is. Cancer is mutated genes that duplicate and then they cause your body organs or processes to not work anymore. All right, quick poll. Changes in DNA as it's copied are always a bad thing. Go ahead and answer that real quick. All right, 5, 4, 3, 2, 1. Come on, people. Got 3 people that have answered that poll. That's not good enough. Let's go. Elijah, you're signed in 3 times. What's going on? When you click on that link.
What you say?
Try clicking on this link.
Oh, I'm talking about the document.
Oh, the document? Are you signed in under your Google? Google?
Yes, sir.
Huh. I'm trying to think what's wrong. Sometimes if you sign out and then sign back in, that might work. Make sure you sign in with your Google on Schoology. And if all else fails, send me a message after class,
Okay.
and then I'll send you a copy.
Okay.
So this is false. Not all mutations are bad. All right, not all mutations are bad. All right, so looking at this, every cell is made up of living things. This is on the right here. This picture is actually pretty awesome when you think about it. So we're going to talk about this piece on the right side, the part where it says DNA double helix. All right, if you kind of follow this, you see these chromosomes that we talk about when cells divide. You see where they split down the center, and then each side has a pair. You know, we're used to seeing the chromosome, but if you think about it, there are 6 feet of this DNA in a chromosome. That's nuts. All right, think about 6 feet tall. It's taller than me. All right, it's coiled up into something that's microscopic. That's insane. All right, just think about that for a minute, and this is how it works. It bundles around these histones, and the histones coil into supercoils, kind of like those old telephones with the long cables that always coil up. That's kind of what happens with the supercoils, and those condense into a chromosome. So this is pretty wild when you think about it. All right, here's a quick video talking about it.
In this animation, we'll see the remarkable way our DNA is tightly packed up so that 6 feet of this long molecule fits into the microscopic nucleus of every cell. The process starts when DNA is wrapped around special protein molecules called histones. The combined loop of DNA and protein is called a nucleosome. Next, the nucleosomes are packaged into a thread. The end result is a fiber known as chromatin. This fiber is then looped and coiled yet again. Leading finally to the familiar shapes known as chromosomes, which can be seen in the nucleus of dividing cells. Chromosomes are not always present. They form around the time cells divide when the 2 copies of the cell's DNA need to be separated.
So you're watching the cell divide right there. So now you got 2 cells.
When DNA replicates,
Okay, so how does DNA code? That is not the right slide. All right, so nucleotides fit together to form base pairs. So the color little slices, tabs on the side of each helix are called the nucleotides. They form base pairs, meaning like this side is a base, it forms a pair with something on the other side, base pair. All right, there in a gene, there's 1,000 of them, but there's 300 base pairs in the human genome. So that's all the instructions to make a human. There are 3 billion base pairs. So there's a lot of this going on inside of you. So a nucleotide has to have a nitrogen-containing base, a phosphate tail, and a deoxyribose sugar. These pairs chemically bond to a nitrogen-containing base pair through chemical attractions, which is kind of like over the core, like what we're trying to cover here, kind of like over what we're trying to do. So what I want you to focus on is adenine pairs with thymine, and guanine and pairs with cytosine or cytosine. So A to T, G to C. A to T, G to C. I'll show you how that works in a minute and why that's important. So I'm going to let you get a second to copy these notes down. All right, so if you're following along, your notes should look something like this. Any questions or concerns so far? Okay. All right, so here's a quick activity. All right, so what I want you to do is I want you to draw in the matching base pair. So I'm gonna need someone to shout this out. What does T pair with? What letter?
A.
A, very good. So anywhere you see a T, you're gonna draw an A. All right, so there's also a T here. A. All right, what does G pair with?
C.
C, good. So I'm gonna put a C here, a C here, and then kind of the opposite, G pairs with C, so a G would go here. So what happens is there's a big protein molecule that slices this in half and copies the complementary base pair here. Now, what happens is in groups of 3, so like say ACC right here, right? That can code for a specific protein. all right? And then that protein can give instructions to carry out somewhere else. So as this DNA is split and paired, these combinations of cells code for proteins that tell your body what to do. So that's why this is important. All right, and you will get one of these later this week for the review at the end of the week, where it'll say, I'll give you a string of base pairs, and you'll have to find the matching base pair that goes with it. So for DNA, A to T, G to C. All right, go ahead and submit that. All right, on a molecular level, this is what it looks like. You have your phosphate tail, then you have your deoxyribose sugar, which is the large portion of it, and then your base pair is attached to the end. So you need a phosphate, a deoxyribose sugar, and a base pair. All right, so the structure of DNA is continued. All right, you have it looks like these rungs of a ladder that's twisted. All right, the sides of the ladder are that sugar phosphate backbone. So it's got the sugar molecule and the phosphate. You can kind of, if you look closely, you can see that thing we just talked about. That blue ball is this phosphate. The hexagon is it's a pentagon is a sugar, the sugar backbone, and then the base pair pops off of it. That keeps it attached to the ladder, and then the base pair bonds between the two nucleotides through hydrogen bonding, which, if you remember way back when in physical science, or if you've taken chemistry, hydrogen bonding is basically, it's a light bond between two atoms based on the pull of their charges of the hydrogens on them. All right, so the rungs of the ladder are the nitrogen-containing base pairs, and the sides of the ladder are the sugar phosphate backbones. So the ladder twists into a helix in order to be compacted down into chromatids. All right, and the last slide for the notes anyways. This happens before the cell can undergo nuclear division. So during it takes the template of a cell is what happens. So before it can undergo division, this stuff happens in the S phase of a cell. You need to make a copy. So we're going to start moving into how, now you've got over the DNA structure, we're going to start moving into the functioning copying of a cell, or I'm sorry, DNA. We're going to talk to more about that later this week, about what specifically happens, but to get you started, you need to take a template. So the DNA splits itself down the middle and uses a template of each side to make a copy of itself. which is where those base pairs come into copy. Because if you take one side and split it in half, the matching base pair will be added to the second half, so you can have two copies. So you need a template strand in order to start this process. So that's that first blank, the template. Now, like when you make a copy on a copier, you use a template document to make the copy of. I'm sorry, I have backwards. This happens in the S phase of a cell, which you can remember is the part where the DNA synthesizes or is made, which makes sense. I put that document little picture on your notes just to give you a reminder because I know it's probably been a while. So the things that are used are DNA helicase and DNA polymerase. All right, so these things can be broken up into base words. So if you see something that says ACE, A-S-E on the end of it, it means that enzymes are proteins that are important in the metabolic process. So that means it's important in metabolizing these proteins. All right, so that should take you through the end of your notes right here. In case you missed anything, this happens during S phase and cell division. DNA uses a template strand using DNA helicase and DNA polymerase. ACE stands for enzymes, which are proteins important to the metabolic process. So here's a picture of where these things come into play. So this helicase molecule splits the DNA down the middle and makes a fork. So you have old DNA is in blue, and then the DNA polymerase comes in and attaches the new base pairs on. So if you split it down the middle, you're going to have those matching base pairs, and you can reconstruct what the DNA looked like based on those base pairs. So like we did in that drawing part, you can reconstruct the old DNA and make a second copy based on its matching base pair. So when this thing, it slides like a zipper, all right? So this DNA helicase, think of it like a zipper. It takes apart the two zippers. And then think of if you took the zipper apart and then there was an additional zipper on top that created a second zipper. So you had a flowing of 4 zippers, which creates your new DNA molecule over here on the other side. Let's see if we have time for this. All right, so let's see. What I will do is I will play this on my shared screen, and I'll let you guys move on to the exit ticket. See, the exit ticket's 3 questions. I'm going to play this in the background while you guys fill out your exit ticket. This is part of the notes for today, so make sure you do it. And it's going to be part of your grade, so make sure you do it. Just to let you know. The exit tickets are based on your participation, not how much you get right. So do your best. Watch the video while you do it. Remember to also turn in your notes when you are done on Schoology. If you can't access them for whatever reason, send me a message on Schoology and I will send you a copy of it. All right, so go ahead and start the exit ticket and watch the video that I had on my shared screen.
State of Theory presents What is DNA and how does it work? DNA, also known as deoxyribonucleic acid, is a molecule. It's a bunch of atoms stuck together. In the case of DNA, these atoms combine to form the shape of a long, spiraling ladder, sort of like this one here. If you ever studied biology or saw the movie Jurassic Park, you probably heard that DNA acts as a blueprint or a recipe for a living thing. But how? How on earth can a mere molecule act as a blueprint for something as complex and wonderful as a tree, a dog, or a dinosaur? To help answer that question, let's first take a quick look at amino acids. Amino acids are tiny little chemicals inside our bodies that are so important, they're often referred to as the building blocks of life. There's about 20 different kinds, each with their own unique shape. The neat thing about them is they can be attached to each other kind of like Legos to produce an endless variety of larger particles known as proteins. Amino acids make up proteins. Proteins, along with other chemicals, combine to form living cells. Cells make up tissues. Tissues make up organs. And organs, when they're all put together and functioning, of course, combine to form living creatures like you and me. These proteins that make up our bodies, and keep in mind there's millions of different kinds of proteins, they each have to be formed in the perfect shape in order to function. If they're the wrong shape, they usually won't work. That's where DNA comes in. DNA does a lot of interesting things, some of which we don't fully understand, but one of its main and most well understood functions is to tell amino acids how to line up and form themselves into the perfect protein shapes. In theory, if the right proteins are built at the right time and in the right place, everything else from cells to organs to entire creatures will come out just fine. This here is a simplified model of DNA. It shows us that the steps of the ladder are made up of 4 different kinds of chemicals shown here by different colors and letters. If you look at just one half of the molecule, you can read its chemical sequence or genetic code from top to bottom, sort of like a book. A single strand of DNA is extremely long, millions of letters long. It spends most of its life coiled up like a noodle, living inside the nucleus or the centerpiece of a cell. Amino acids, however, live outside the nucleus in what's called the cytoplasm. To help DNA interact with the cytoplasm and convert those amino acids into proteins, special chemicals inside the nucleus make partial copies of the DNA code. These partial copies, called RNA, look a lot like DNA, but they're shorter, of course, and they're missing one of their sides. Their small shape and size allows them to fit through tiny pores in the nucleus, out to the cytoplasm, and into the mouth of another particle called a ribosome. Ribosomes are protein-building machines. They read the RNA code 3 letters at a time, suck amino acids out of their surroundings, and stick them together in a chain according to the RNA code. As the chain grows, it bends, it folds, and it sticks to itself to form a perfectly shaped protein. Every 3 letters of the RNA code tell the ribosome which of the 20 different kinds of amino acids should be added next. For example, CAA tells the ribosome to grab a glutamine. AGU tells it to grab a serine, and so on. Once a protein is built, it can then go on to do a number of different things, one of which could be to help form a brand new cell. So to answer the original question, what is DNA? DNA is a molecular blueprint for a living thing. How does it work? DNA creates RNA, RNA creates protein, proteins go on to form life. This entire process as complicated, as sophisticated, as magical as it might seem, is entirely based in chemistry. It can be studied, it can be understood. I'm John Perry, and that's DNA stated clearly.
Okay. So make sure you go through and you answer the exit ticket that's on near pod. It's on the exit ticket is on near pod. All right, once you are done with the exit ticket, you are free to go. Remember that I'll be posting these lectures on a new YouTube page. I'll post in Schoology here in a little bit. So once you are done, you are free to go. Make sure that you do the exit ticket. It is part of the notes for today, and submit your notes on Schoology. If you have any questions, drop them in the chat or send me a message on Schoology when you're done. And once you guys are done with the exit ticket, you're free to go, and I will see you guys tomorrow.